Chapter 6 covers chromosome level mutations. Now, we are starting big and we will work our way to much finer, smaller scale mutations that affect the expression of genes and the specific products that are produced by those. And those are the mutations with which you are probably more familiar. These are the changes in single nucleotides, insertions, deletions, silent mutations, synonymous mutations. These are terms that you have heard before. What we're learning about right now are mutations that affect the genome writ large. And so by this I mean we are changing the number of chromosomes or affecting, by let's say deletion, a very large chunk of a chromosome. So we are working on considering very large-scale mutations in this chapter. And so these mutations are generally far more substantial, as I've said, than those that produce the different alleles for different eye colors in humans, for example. And these might take the form of a deletion of a portion of a chromosome or the duplication of a portion of a chromosome. That's a really interesting instance of a chromosomal mutation. And then other rearrangements that are possible through the movement of genetic elements from one chromosome to another. Some really interesting things that can happen. Now we're going to start by discussing variation in the number of chromosomes. And there are two broad categories of genetic mutations that result in variations in the number of chromosomes within an organism. The first of these is the addition or loss of a subset of the chromosomes that an organism possesses. So let's think about what that means for a moment. You possess 46 chromosomes in every one of your cells. Your father gave you 23, your mother gave you 23. So in total you have 46. This addition or loss of a subset would manifest itself in the presence of, let's say, 45 rather than 46, or 47 rather than 46, okay? This is not a whole another set of 23 or one fewer set of 23. That is this second category here in which we might observe one or more haploid sets being added or, less frequently, removed from the genome of an organism. So the first of these, the addition or loss of a subset of chromosomes, is referred to as aneuploidy. And I want you to think of aneuploidy as an imbalanced number of chromosomes. Okay? You have 46 chromosomes, that's pretty balanced. You can give one of each of those 46 to your offspring. You don't give them 2 copies of chromosome 1, 2 copies of 2, 2 copies of 3. You give them one of every single one. You give them 23 chromosomes, okay? Because it's balanced. Everything your mother and your father gave you, you give your offspring one of each of those things, okay? Just as your parents gave you one of each, okay? The addition of one or more haploid sets is a change in the total number of chromosome sets that exist within an organism, okay? And this is referred to as euploidy. Now, aneuploidy is almost certainly the condition, or one of the genetic conditions that you're probably most familiar with, okay? And we'll talk about which one that is here in just a moment. But right now, what I want to focus on is 2 chromosomes. These are 2 homologous chromosomes, meaning they have the same genes in the same order, presumably. One of these chromosomes came from mom, one of them came from dad, okay? Now, for you, you should have 23 pairs of chromosomes, one from mom, one from dad. And let's ignore the 23rd pair, the sex-determining chromosomes, focus on the other 22. Right? These were given to you by your parents, and you have 2 of each. You are disomic, okay? Now, an instance of aneuploidy would be an individual that is monosomic for a particular chromosome, okay? This is not a balanced number. Clearly, they received a copy of this chromosome from one parent, but not from the other. Okay? This is imbalanced because this individual is incapable of producing equally balanced gametes. Okay? So look at that box at the top and think about what happens when that organism produces gametes and what ends up in every one of those gametes. Every one of those gametes is going to have an orange or a blue copy of this chromosome, either a paternal or a maternal copy. Okay? If on the bottom left we're looking at a cell and one of the chromosomes in that cell, if that cell were to go through meiosis, some of those gametes would get an orange copy and the others get nothing. Okay? So this is an imbalanced number in that balanced gametes cannot be produced. Trisomic individuals, on the other hand, the other end of the aneuploid spectrum, right? Remember, aneuploidy is the addition or loss of a subset of the chromosomal sets or the haploid set. And so here, this trisomic individual has 3 copies of this chromosome. Looks like they got 1 from dad and 2 from mom, okay? And this, too, is imbalanced because think about for this homologous chromosome, they've got 3 copies of it. How do they split that equally amongst their gametes? It's impossible. It's not a balanced number, can't be split, okay? The example within humans that you are likely most familiar with is trisomy 21 or Down syndrome, okay? And Down syndrome is an exceptionally common genetic disorder formed by what we call a non-disjunction of chromosome 21 during meiosis. And we'll get to that in more detail in a moment, okay? So this is an imbalanced number. If I tell you to take this cell that contains all of these chromosomes and run it through meiosis 1 and meiosis 2 to produce a bunch of sperm, okay, you would tell me that you expect half the sperm to get a Y, half the sperm to get an X, half the sperm to get that chromosome 22 on the left, half to get the one on the right. But 21 is difficult. It throws off the whole meiotic division and the allocation of chromosomes. Okay, so now let's consider the other mechanism whereby large-scale chromosomal mutations occur, not producing this imbalanced number, but a change in the number of chromosomes that is effected by the addition or loss, most likely the addition of full sets of chromosomes, okay? So you and I are both diploids. We have 2 copies of everything, one from mom, one from dad, but that's not the case for all living organisms. There are organisms out there that are triploid, that are tetraploid, so they might have 3 copies of everything or 4 copies of everything, okay? In the instance of a tetraploid, there are a number of species of frog. One of them lives here in Mississippi, one of the gray tree frog species that is actually tetraploid. And so when a tetraploid gray tree frog produces sperm, it puts 2 chromosome number 1s in it and 2 chromosome 2s. And when that sperm meets an egg from the same species that also has 2 copies of 1 and 2 copies of 2 in it, it forms a 4N zygote, a tetraploid zygote. Notice I used the terminology 4N, okay? N is the haploid chromosome number. For us, that's 23. You, being a diploid, are 2N. You have 2 times 23. These gray tree frogs are 4N. They have 4 haploid sets of the chromosome. These are the different states of euploidy. And table 6.1 clearly indicates mathematically how you would recognize these different forms of aneuploidy and euploidy. Now, for the most part, the aneuploid systems are not terribly difficult to recognize, right? The difference between monosomy and trisomy, okay, trisomy 21, they have 2 times 23 plus an extra chromosome 21, right? Instances of monosomy would be illustrated by individuals that have 2 times n, 2 times 23 is 46, minus 1, they'd have 45 chromosomes. Okay. These others, tetrasomy, pentasomy, these are far less common, but you can see how this is written. In euploidy, again, these are multiples of n being the haploid chromosome number or set, a chromosomal set. You are diploid, but there are a number of organisms that are polyploid. Poly just means more than 2. And so for each of those polyploids, there's a specific term. There's a term for 3n, 4n, and 5n, as you see below. But they're all categorized as polyploids. So there are 2 other terms down there, autopolyploidy and allopolyploidy. We're going to get to those here in a second. But I want you to think about how these aneuploid chromosome numbers can arise. How do we end up with trisomy 21? How does a gamete form that has 2 copies of the same chromosome? That shouldn't happen, right? They should have been torn apart in meiosis. Well, it happens through a process called non-disjunction. And so what happens in non-disjunction is that these chromosomes fail to separate during meiotic division. And I've got an illustration here of how that might happen during meiosis too. Okay? And now in metaphase, as you might expect, these chromosomes line up on the metaphase plate, and there are microtubules attached to either side of these replicated chromosomes. And each side of these chromosomes, each of these sister chromatids is of course identical to the other. And what is to happen in anaphase too is that each of these replicated chromosomes is pulled apart, and the 2 daughter chromosomes go into what will then become 2 different gametes. But you can see in the instance of non-disjunction for this red chromosome, what happens is the replicated chromosome does not get pulled apart. Okay? All of it goes to the left. And what we are going to end up with is a chromosome, or 2 gametes, because this is anaphase 2. We're going to end up with 2 gametes after this. Each of them has 1 copy of the blue chromosome, 1 of the green, but the left gamete is going to have 2 copies of the red chromosome. The right gamete has only 1 copy. Let's say both of those gametes fuse with another gamete and create a zygote. The one on the left is going to be trisomic for the red chromosome, right? Because there's going to be a 3rd copy of the chromosome that comes with the other gamete. The gamete on the right is going to be monosomic. It brings 0 red chromosomes to that zygote. Presumably the other gamete does bring a single copy, and therefore we have monosomy. And so it's through this process of non-disjunction that aneuploid genotypes are formed. Now, non-disjunction doesn't necessarily have to happen in meiosis 2. Okay? You can have non-disjunction in either the first division, so meiosis 1, or in the second division in meiosis 2. So let's look here what happens. We're starting with, at the top, 2 homologous copies that have been replicated of the same chromosome. Okay? And after the first meiotic division, if instead of one of those going to the right and one go to the left, they both go to the left, we've got a problem. Okay? Because when that cell undergoes the second meiotic division, all that happens is each of those 2 replicated chromosomes is pulled apart, okay? With half going to the left, half going to the right, and you're going to end up with both of those gametes having 2 copies of that chromosome from the single parent, okay? And if it fuses with a normal haploid gamete on the bottom left, you end up with trisomic individuals, okay? Conversely, on the other side of this, you can see that the cell that resulted from the first meiotic division, the one that did not receive any copies of this chromosome, goes on to divide. The gametes have 0 copies. The other gamete brings with it a single copy, so those zygotes would be monosomic. Okay. If disjunction, on the other hand, is normal in meiosis 1, as you see here, but there's a non-disjunction event in meiosis 2, okay, in that this replicated chromosome does not get pulled apart. Both halves of this replicated chromosome go to the left, none go to the right. You'll see that the two gametes that are formed on the far left through normal disjunction will produce disomic normal offspring, right? But the two gametes on the right, the one on the left, will produce trisomic offspring, the one on the right, monosomic offspring. So you can see here that non-disjunction can act in both meiosis 1 and meiosis 2 and result in aneuploid offspring. So what are the effects of aneuploidy? Well, monosomy is frequently lethal, okay? It's most often lethal, and there are few instances that I'm aware of other than the Turner syndrome, in which offspring have only one sex-determining chromosome, a single X, okay? That's the only instance of monosomy that humans survive. So this is a very lethal condition in humans, but it can occur in other species. And the reason it's believed to be so detrimental to the well-being of the individual that bears this mutation is due to something called haploid insufficiency. And basically, this is exactly what it says. If you are haploid, if you have only one copy of a chromosome and all the genes on it, that is insufficient for the production of all of the gene products that are encoded on those chromosomes, okay? Now, trisomy, this is far more common in humans. For the larger chromosomes, this is almost always lethal, okay? There are 3 instances, Patau syndrome, Edwards syndrome, and Down syndrome, the latter one you are more familiar with. Those 3 instances of trisomy, so trisomy 21, 13, and 18, all 3 of those individuals can survive, but they have serious malformations or other issues, as you are likely aware, okay? So, how frequently do these chromosome level mutations occur? Well, it turns out that studies of lost pregnancies suggest that while about 20% of pregnancies or conceptions terminate in spontaneous abortion, about 30% of all of those have some form of chromosomal abnormality, which is a pretty remarkably high number. So that just gives you an idea of how commonly this occurs and how detrimental it is to have one of these chromosome level mutations of aneuploidy. Now, polyploidy, this is the other side of the coin, the euploidy side of the coin, in which we've got organisms that have more than just the 2 copies of each chromosome. This is incredibly common in plants, far less common in animals. The gray tree frog example I gave you is one of the few that I'm aware of in vertebrates. And it's partly because this is a really significant change in the genetic composition of an organism, okay? And it arises, as you might suspect, much like aneuploidy, due to segregation issues during meiosis. But it can also be the product of hybridization among different species, and we'll get to that in a moment. So you'll remember when I showed you table 6.1 that at the bottom I mentioned auto and allo polyploidy. We're going to dig into what those are right now and what that means. In an auto polyploid, if you start out with a diploid indicated by an organism at the top, so that is 3 chromosomal pairs, homologs lined up next to one another. This organism would be 2n equals 6, n equals 3. So they have 2 of all 3 chromosomes. They got 1 from each parent. Now, an autopolyploid is a polyploid species that could be triploid or tetraploid, that has multiple copies, more than 2 sets of the chromosomes for that same species, for one species. So basically a tetraploid for this species would have 4n equals 12. They have 12 chromosomes total, bottom right here. They have 4 of each of the 3 chromosomes, but you'll see that all 3 of those, so all 4 of each of those 3 are the same. They look the same. They've got the same genes, the same order, everything's the same. They just have 4 instead of 2. A triploid has 3 instead of 2. Same thing. Auto simply means they have multiple sets, more than 2 sets of the same set of chromosomes. Okay? So it's as if you might have, instead of 2 times 23 is 46, you might have 3 times 23 is 69. Okay? An allopolyploid is very, very different. And in this, what we have is an organism, the tetraploid at the bottom here, who exists with a genetic composition that is derived from 2 different species. And what they have is the full diploid set from the orange species, and they have the full diploid set from the blue species. You can see it is intentional that these chromosomes were illustrated to have different sizes and shapes and orientations because they come from different species. They have different genes on them. They are very different chromosomes, very different genomes. And so what this tetraploid is basically a fusion of both of these organisms. It has all the genes of the blue species, all the genes of the orange species. It has the full diploid set of both. And this is quite a remarkable situation. This almost never occurs in animals, okay? But it is quite common in plants. And I bet you're thinking, I hope you're thinking, how on earth does this happen? How do 2 diploid organisms like this, the blue species and the orange, produce gametes that create a tetraploid offspring? Well, it happens through the abilities of plants that are a bit beyond ours. Okay, so consider here 2 different species, 1 and 2. Okay, species 1 has 2 copies of 3 different chromosomes. Okay, this species 1 is 2n equals 6. Okay, and when they produce a gamete, that gamete would have a copy of chromosome 1, a copy of chromosome 2, and a copy of chromosome 3. Right? Now the other species, number 2, has 2n equals 4. They have a chromosome number 1 and a chromosome number 2. When they produce a gamete, you would get a B1 and a B2. Okay? Now what's remarkable in plants is that it is possible that these 2 come together and one fertilizes the other. And you end up with a zygote that has a copy of A1, A2, A3, and B1 and B2. Now, if this is as far as it went, you would end up with, as it says here in the figure, a sterile hybrid. That cell cannot undergo meiosis and produce anything that resembles a balanced or organized gamete, right? Think about this cell going through meiosis to produce a sperm, okay? What would be in the sperm? An A1 and a B1? Or an A1 and an A2 and a B2? There's no way to divide these 5 chromosomes and split them into cells. But what plants are capable of doing is actually chromosome doubling, which is shocking. So this is a really interesting thing that plants are able to do. And we can actually do it in the lab. We can facilitate it by the administration of some chemicals that sort of kickstart this process. But chromosome doubling means that that sterile hybrid that had only 1 copy of all 5 of these chromosomes can say to itself, and of course this is not an actual thought of the plant, but what the cell does is recognizes this insufficiency of having only 1 of everything, and it simply goes in and makes another copy of each. So they have 2 A1s, 2 A2s, 2 A3s, 2 B1s, 2 B2s. that cell can undergo meiosis and produce gametes. It could produce gametes that have what you see down on the bottom, an A1, A2, A3, B1, B2. Okay? This fertile allopolyploid now can produce gametes that if you put those two gametes together, you'd produce something just like the parent, right? And so these allopolyploids can sometimes be referred to as an amphidiploid. Okay, your text talks about that and when that applies. Basically, it applies when we know what the two parental species were in the first place, what species A and what species B were. Okay? In this allopolyploid example, we have a species that instead of having 2 times 3 is 6 or 2 times 2 is 4, this species now has 2 times 5 is 10. Can this species breed with either the A or the B? Could you put any of these gametes on the bottom with the gamete that would be produced by a species A or a species B? No way, okay? Those gametes would not be complementary. And so in creating this hybrid, this amphidiploid hybrid or this allopolyploid hybrid, if it goes through chromosome doubling and produces gametes, guess what? Those gametes can only fuse with the gametes of another allopolyploid of the same background, okay? And so this is an instance in which you can instantly create a new species that cannot breed with either of the parental species. It's a super cool situation. This has actually been documented to be how we come to have the species of cotton that is grown here in the United States. It turns out that there's a native species of cotton in the United States, a native species in Europe. The European species has 13 small chromosomes. The native North American species has 13 large, okay? And what's remarkable is that the cotton that we grow has 26 chromosomes, okay? And this number is the result of the fusion of gametes from these two different species and then chromosome doubling. It's quite remarkable. All right, so let's think now not about affecting the number of chromosomes, the quantity of chromosomes, but mutations that affect the composition and arrangement of the chromosomes. And I'm going to go through some very simple examples. These are very well illustrated. There's not a lot to think about here. It's just understanding the different types of mutations and what they're called. So in looking at these, we're dealing with a chromosome, a linear chromosome that has genes A, B, C, D, E, F, G, and in that order, okay? Now mutations to that chromosome could be mutations in which a chunk in the middle disappears, or it could even be a chunk at the end. It doesn't really matter where it is. But if some of that chromosome is lost, it has been deleted. So a deletion, in this case, we have a deletion of the region in and around gene D. You get a shorter chromosome and one that does not have gene D on it anywhere. The next one illustrates a duplication event in which the region in and around genes B and C is actually duplicated. So now the chromosome's longer and you have gene B twice and C twice. Inversions, you simply have chunks of the chromosome that are flipped around. Their orientation is changed. And you can see here that they are now out of order with respect to where they were at the beginning. Terminal deletions, you can just have a break or a chunk of the end of the chromosome disappear. This is actually indicated in one of the genetic maladies that affects humans, Creutzfeldt-Jakob syndrome. This is an interesting genetic disorder in which a portion of chromosome 5 is missing. So what's remarkable is that these are humans that survive they are missing only a very small portion of the tip of chromosome number 5, just one of them. They have another chromosome 5 that's complete and whole from one of their parents, but one of them they received is short. And again, this highlights the haploid insufficiency or haploinsufficiency that we mentioned earlier, in which you need 2 copies of most all of your genes in order to produce enough of everything and be viable. Your text digs into how these deletions might happen in the middle of these chromosomes, how chunks might be lost from the middle. So this is just the difference between a deletion that is either terminal at the end or intercalary somewhere in the middle. Okay, it's not terribly difficult to understand, nor is it a critical concept that you understand, but just be aware that deletions can take place in multiple places throughout the chromosome. And just understand that when these chromosomes align to one another during meiosis, that deletions form a problem for the alignment of homologous chromosomes. So when these 2 line up, if they have different lengths and one of them has a chunk of it deleted from the middle, you might end up with some issues of alignment. Okay, so a portion of this blue chromosome that is longer and not present in the yellow chromosome has to be looped out so that the homologous regions can align to one another. Okay, so translocations. Translocations are an important category of chromosomal mutations in which chunks of chromosomes get moved to non-homologous chromosomes. That's the important point here, is that this is not crossing over, as you've learned about it in 160 and in other classes. This is the exchange or the movement of a chunk of one chromosome to another non-homologous chromosome. So this exchange or translocation can be reciprocal or non-reciprocal. Okay? And you've probably heard this term before, or you've heard the term reciprocate. If you reciprocate for someone, that means they do something for you, and you reciprocate, you do something back for them. Okay? In a reciprocal translocation, that's exactly what happens. You can see here that these 2 chromosomes that are not homologous, you can tell they're not homologous here, the blue and the orange, because they are different sizes and they have different genes on them. They undergo a translocation in which they get tangled with one another and exchange a portion of their chromosomes with one another. And after this translocation, genes H, I, and J are attached to and connected with C, D, E, F, G. Okay? And A and B are with KLM. This is a scenario that is only created when non-homologous chromosomes exchange portions of themselves with another. Now, a non-reciprocal translocation, you can see on the left, is simply when a portion of one chromosome gets moved to a non-homologous chromosome. Okay? And so what happened here is genes A and B, that portion of the blue chromosome, broken off and was attached to the top of the orange chromosome. There was no exchange, it was just a unidirectional movement, and so it is non-reciprocal. Okay. Now, we can also see how these types of exchanges can happen with homologous chromosomes, and sometimes those exchanges are also not reciprocal. Okay. So this might be an instance during meiosis 1 when these homologous replicated chromosomes line up next to one another. Look on the left. The replicated blue chromosome and the replicated orange chromosome. They have the same genes on them, but they've gotten tangled with one another, if you will. And what ends up happening is they swap their distal ends, their top ends, and what moves to the left, to the top of that blue chromosome, is genes A, B, and C. But what moves to the right on the top of that orange chromosome are genes A and B. And so what happens here is not only is this exchange not exactly identical, they're not swapping identical regions, but look at that chromosome number 3. It has 2 copies of gene C now, just because this exchange was not perfect. And so we have the duplication of a gene. And the duplication of genes is a huge deal evolutionarily, okay? Because genes that have been duplicated, when you have multiple copies of a very important gene in your genome, I want you to think of that as basically a mutational freebie, okay? Think about it this way. If you have to get to class every day and your class is at 7 a.m., there's no buses that you can get where you live, you've got to have a car to get to class on time, okay? If that's your schedule, okay, and you've only got one car, if that car breaks down, you're screwed, okay? If you only have one copy of a gene and it is mutated, it suffers a mutation, you're screwed. You can't produce that gene product. But if you have 2 cars or 3 cars or 4, one of them breaks down, no problem. The same is true of gene duplication. And so here's an example showing the number of copies of both the alpha and beta portion of your hemoglobin gene. So basically you have 2 different genes, an alpha and a beta. You make 2 different proteins, alpha and beta, and they get stuck together to make your hemoglobin. What's crazy is you have 6 copies of the beta and 5 copies of the alpha. And so notice that 3 of these, 1 of the betas, 2 of the alphas, are indicated to be pseudogenes. What that means is they don't work. If you make that gene product, it's not a functional beta or a functional alpha subunit of hemoglobin. Doesn't work, okay? Why doesn't it work? Because there was a mutation that broke them after they had been duplicated. What's the benefit of having 5 functional copies of beta globin? Well, if there's a mutation to one of them, you and or your offspring can still survive because you've got 4 other copies that produce functional beta globins, okay? The other important aspect of this is that this is how evolution of a gene that is incredibly important to the fitness of an organism can proceed, okay? You can't have mutations to something that change it. It's too risky and problematic if it has the potential to make that gene product unavailable and unviable, okay? So having multiple copies opens up mutational potential. Another aspect of copy number variation is that the number of copies of a gene in the genome can be directly correlated with the quantity of that protein that you can make, okay? So just think about the fact that if you need to produce something, if your body needs to produce something, you go to that gene and transcribe and translate it. We'll be learning about that. But you make that protein. Well, if you only have 2 copies, 1 on the chromosome from mom and 1 on the chromosome from dad, you can produce a set amount per cell. But what if you have 10 copies on each chromosome? Well, you can produce 10 times as much. And this has been shown to be the case in a number of instances in which organisms or species vary in their phenotype. I have a rattlesnake here because there are some populations of rattlesnakes that have much more toxic venom than others. And in some instances, this has been demonstrated to be due to the fact that some populations have more copies of the gene that, when transcribed and translated, produces this toxic protein. So the more copies of the gene you have, the more toxic your venom is. A pretty remarkable mechanism for the evolution of toxicity. Okay, so another aspect of genetics mutations at the chromosomal scale are translocations. And I already touched on this a little bit, but this is changing the position of a chromosomal segment. And so here you can just see the uh, a reciprocal translocation, okay, between non-homologous chromosomes, creating new combinations of genes. Okay. I just wanted to touch on this again because there's an important example that I want you to be aware of, in that one of the genetic conditions that you're most familiar with, Down syndrome, as you know, is produced by having 3 copies of chromosome 21. But there's more than one way to get there, and there's a really unusual way to arrive at trisomy 21 that was discovered when researchers observed that some families had a higher incidence of Down syndrome than you would expect by chance. And this turns out to be a condition called familial Down syndrome. And it's due to a translocation of a portion of chromosome 21. Let's take a look at what that looks like. And so on the left, you can see a normal individual that has 2 copies of chromosome 21, 2 copies of chromosome 14. And when they produce a gamete, that gamete, bottom left, has 1 of 14, 1 of 21. But what's interesting is that individuals in these families that were producing high numbers of offspring with Down syndrome, these individuals had a normal phenotype. They looked normal. They did not have Down syndrome. But when their chromosomes were examined, it turns out that they only had 1 chromosome number 21, which is weird. They shouldn't have been able to survive. But when looking at chromosome 14, it turns out that one of their chromosome 14s is much longer than would be expected. And it turns out that's because they have a translocation. A big chunk, the vast majority of chromosome 21, has been stuck on to chromosome 14. And so this individual, look at that karyotype at the top middle, or that chromosomal composition. They have a normal chromosome 14, they have the blue one, they have a normal chromosome 21, the orange one, and then they have that one in the middle. It's mostly blue and partly orange. What that is a chromosome that is a fused 14 and 21. It's got pretty much everything from 14, it's got pretty much everything from 21. That individual effectively has 2 copies of chromosome 21 and 2 copies of 14. They're fine. That's why they don't exhibit any symptoms of Down syndrome. But when they produce gametes, take those 3 chromosomes and you run it through meiosis and see what happens. And you will see that the 4 gametes at the bottom are all that can be produced from the meiotic division of a cell that has only these 3 chromosomes. Okay? And when you fuse those with gametes from another individual on the bottom left there, you'll see that 25% of the offspring are going to be normal. Okay? 25% will be just like that carrier parent. They have the translocated 14-21 combo. 25% are going to have Down syndrome, but they have Down syndrome not because they have 3 chromosome 21s, they only have 2 of them, but one of their 14s has that extra chunk on it. So effectively, they have 3 copies of all the genes on chromosome 21. And then 25% of the offspring are monosomic for chromosome 21, and that is lethal. So you can see here a karyotype of a normal Down syndrome individual, not familial Downs. They have 3 copies of chromosome 21. But if we consider what these translocation carrier genotypes look like, or what an individual with Down syndrome that has familial Downs, here is their 2 copies of 21, their normal 14, and then this fusion 14, showing clearly that they have 3 copies of chromosome 21 effectively. All right, the last bit we touch on in this chapter are fragile sites. And this is, surprisingly, the most common inherited intellectual disability in humans. And you can see a portion of this X chromosome here indicated by an arrow, in which dyeing and staining has clearly indicated this fragile site. And what's interesting is that this is a region of the X chromosome that is susceptible to breakage. And I put that in quotes because I don't necessarily mean to imply that part of the chromosome falls off. It's broken in that it no longer functions properly, okay? And so studies suggest that when individuals have the genetic composition that leads to this Fragile X syndrome, this breakage, in the absence or with low levels of folic acid present, okay, they can lose the function of this portion of the X chromosome, which can cause serious problems. And what causes this breakage is a repeated region of nucleotides. So you'll remember the chromosomes are made up of G's, A's, T's, and C's, all strung in a row, okay? And some of the chromosomal regions actually contain information. They're genes, and that information is used to code for a particular protein. Now, not all of that information is used to code for information and is not part of genes. There are lots of regions that just have what appear to be randomly repeated junk. And for humans, in this region of the X chromosome, there's a bit or a region in which you will find CGG repeated. I might have it 6 times, you might have it 8 times, the person down the street might have it 92 times, okay? If you have somewhere in the neighborhood of 6 to 54 repeats of this CGG in the region of this FMR1 gene, you're fine, okay? But if you have more than 230 repeats of CGG near this gene, what it effectively does is shuts off this FMR1 gene, resulting in the mental handicap that was described, okay? And so this is digging into a bunch of things that we're going to be talking about, notably the function of genes, what they produce, and also these repeated regions in the genome and how they can affect things, okay? But I simply want you to understand what these fragile sites are and what can cause them, notably these long regions of repeats that can inactivate the expression of a particular gene. Okay, so that's all for chapter 6.